Okay, so welcome everyone to this MAPP round. My name is Vincent Maloto. I'm a postdoctoral research fellow here at the Familial Dementia Neuroimaging Lab and the Multicultural Alzheimer's Disease Prevention Program. It's my great pleasure today to introduce Professor Bernard Hanseeuw from UCL Louvain in Belgium. Professor Hanseeuw is a behavioral neurologist conducting research on Alzheimer's disease with the aim of better caring for patients with or at risk for memory decline. He obtained his MD from UCL Louvain in 2007 and a PhD in neuroscience from the same university in 2012. After being board certified in neurology in Europe in 2014, he realized a 3-year postdoctoral fellowship in Reza Sperling's and Keith Johnson's lab at Harvard Medical School. And since he moved back to Belgium, he kept the position of part-time instructor at the Gordon Center for Medical Imaging. Professor Hanseeuw is now the co-director of the Memory Clinic at Saint-Luc Hospital in Brussels and the president of the Belgian Neurological Society. His research is supported by the Belgian National Fund for Scientific Research and the Belgian Foundation for Alzheimer's Research. His main research interests lie in the longitudinal assessment of amyloid and tau pathologies in clinically normal and mildly impaired older adults, and the biochemical study of tau in human biofluids and brain samples. Today's talk will be focused on one of his latest research projects involving the analysis of post-mortem human brain data, titled Specific Modification of the Tau protein distinguished between Alzheimer's disease and primary tauopathies. So I will now give him the floor, but please feel free to type in your question in the Q&A during the presentation, and we'll go through them at the end of it. Thank you.
So thank you very much, Vincent, for this very nice introduction. Thanks to Yakel for suggesting my name for this MAPP seminar. I'm very happy to be with you today. I will talk about neurodegenerative disorder. Based, first of all, on my clinical experience that is guiding all of my research. And so I've spent 3 full full-time year of research in Boston, but I have never seen patients there. I have my clinical experience in Brussels in the memory clinic. That is a free access memory clinic where we see 2,500 patients per year. Among those, around 75 are diagnosed with MCI every year, and we include most of these in research and academic studies or clinical trials. Since 2019, so 5 years ago, I have reinstated the brain donation program, and that has allowed us to collect brain samples and to initiate a collaboration with the Netherlands Brain Bank in Amsterdam that has way more many brains since they have started in 1985. So to just start with a broad overview, we can think of neurodegenerative disease as a crime to solve. What is the crime? The crime is the crime of or cognition or memory, or in some rare disorders of the motor function, one of the brain function. And the crime that we need to solve is basically what is the biological culprit? Who is responsible for this crime? One tool that I have used very early in my research career are tools assessing brain dysfunction, either with functional MRI or with FDG PET, as you see here on this map, where you see in blue regions that are dysfunctioning. Now, what is very complicated in Alzheimer's disease is that we do not think to have only one culprit, one person responsible, but at least 2. I have named the first one the killer. The killer is a tau protein. And as you can see on this tau path, we find the killer at the exact place where the crime is committed. It is not possible to have a crime if the killer is not at that place at that time. But now we have another culprit or another responsible protein that is called amyloid. which in my comparison is the order. The order of a crime can be given many years in advance. And the person giving that order doesn't need to be where the crime happens. So this is what we see with amyloid PET imaging, where we see amyloid aggregates depositing in many different brain regions, including brain regions that are functioning well. And amyloid deposits in the brain many years before the brain starts dysfunctioning. The problem when you are a researcher and you have limited research capacity is that you have to prioritize what you're doing. Just the same way that when you are the policeman trying to solve the crime, you have to prioritize who are you going after. And historically in the field, there has been much more effort on amyloid than on tau. Basically, scientists want to understand the primomovens, the prime protein dysfunctioning. Who was responsible for that crime is the person who gave that order. Now, to keep the comparison, one issue with that is that you have a risk to put in jail every man that once wanted to kill his mother-in-law. But maybe not all those men were going to actually kill their mother-in-law. So maybe we're putting into jail many people that would never commit a crime. Going back to the Alzheimer's disease or the neurodegenerative disease comparison, we risk to treat and to consider as diseased many individuals that will not experience dementia in their lifetime. And putting the accurate focus is very difficult in this sequence. Now, PET imaging, as you see on this slide, is very accurate to measure where in the brains the disease process is occurring and at what time, because PET imaging, in contrast with autopsy, allows to be repeated and to observe the sequence longitudinally over time. Because PET imaging allows to visualize two aggregates in the brain, and those aggregates are depositing in the brain regions that start dysfunctioning, it has been very important to develop tools allowing to visualize this extent, this expansion of taupathology in the brain. And that's what you see on this slide, where we can measure the percentage of voxels that are above a certain threshold. And we have tried back in Brussels to dissociate this measure of extent, symbolized here by extent of tau, EOT, the percentage of voxels that are positive, from the SUVR, which is a typical PET measure, measuring signal intensity. How high is the signal? Basically, one is shown on the upper line, the other one on the line below. You lose the intensity in the line below. And what we could demonstrate is that this metric, the spatial extent of tauopathy, shows a strong association with cognition that the signal intensity in patients with MCI due to Alzheimer's disease, or even in clinically normal individuals with Alzheimer's pathology. Now, pushing further those PET work, we have compared, and Vincent has maybe already explained this work to you because it was part of his PhD. We have compared two PET images and brain function images to start suspecting the presence of non-Alzheimer's pathologies. When would we suspect such a pathology? Well, when we see brain dysfunction in a region that is not affected by tau, which is basically a suspicion that another proteinopathy or another brain disorder is responsible for dysfunction hypermetabolism in this region. A last piece of work that I wanted to highlight using PET is this recent paper from the Harvard Aging Brain Study, where I had the honor to look at hippocampal atrophy once tau and amyloid had been adjusted, and where we could observe that hippocampal atrophy was responsible by itself for 10% of cognitive decline independently of amyloid and tau, suggesting again that other processes are at play. So the reason why I developed such an interest in tau pathology is because tau is more proximal to brain and cognitive dysfunction than amyloid is. It's my clinical hat. When we are seeing patients, most often they already have tau pathology when you diagnose Alzheimer's disease. Even at the MCI stage, most of them have at least Bragg stage 1 or 2. The next question to me is, how do amyloid positive clinically normal individuals develop tau pathology? Going back to my comparison, this is the question of who is the messenger? How is the order transmitted to a killer? To me, someone who wants to commit a murder is not guilty until he actually sent a message for the crime to be committed. And between amyloid and tau, there is a lot of biochemical research, but the links are still not well understood. And PET is not the best tool to understand those links because PET doesn't describe the biochemistry. PET describes very accurately the place, the brain regions where those aggregates develop, the sequence, the temporal sequence of development, but not the actual biochemical signals that those pathologies carry along. 6 years ago, I had the chance to meet with a biochemist named Nicolas Barthélemy, who was convinced by then in the power of tau biochemistry to describe the sequence of events in Alzheimer's disease. He was working at Washington University in familial Alzheimer's disease, a cohort that you are all probably very familiar with, the DIAN cohort. And he could demonstrate that tau phosphorylation occurs on different epitopes, so basically on different amino acids, at different time in the sequence of development of Alzheimer's, of autosomal dominant Alzheimer's disease, with tau on threonine 217 being the first one to be detectable around 21 years before dementia onset. And another phosphoepitope, the phosphoepitope threonine 205, occurred a little bit later, around 13 years before dementia onset, which is about 5 to 8 years before MCI. Of note, the threonine 205 site is the antibodies that is used by neuropathologists to define Bragg stages, so basically to detect tau tangles. In a subsequent work, he could show that all these epitopes correlate with tau PET, but some of them better than others. For instance, threonine 181 that has been largely used in clinic because it is the historical measure in CSF is moderately correlated with tau PET, and we have reproduced similar results in tau data from both HAPS and the clinical cohort we have in Belgium. In contrast, 217 has stronger correlation, but the best epitope that was looked at in this study to predict tau PET signal was the threonine 205, which makes sense if we think that that is the one that is associated most with tau aggregation in the brain. But all these phosphoepitopes have now been largely studied in the spectrum of Alzheimer's disease. However, we know that non-AD tauopathies are frequent in the elderly. They are not as frequent as AD, but they are frequent either as a sole pathology, and that's pretty rare, or as co-pathologies as well. So a brief reminder of tau pathology and all tauopathies that are observed in the elderly. The most common tauopathies is called 3R4R. Where does that name come from? It comes from the isoforms of tau. Tau is binding to the microtubule, is the function of this protein to stabilize it. And some proteins have 3 repeat sequence to bind to the microtubule, and others have 4. The biological reason for that variation is that 3R tau doesn't bind as well as 4R tau. And sometimes we need some flexibility in the brain to be able to move the neurons, to move the axons, and to create new connections. And so, for instance, in the fetus brain, the human fetus brain, there is only 3R tau and no 4R tau. In the adult human brain, we have a mix between both. And in Alzheimer's disease, we see both types of isoforms aggregating in the brain. It is the same type of aggregates that we see in primary age-related tauopathy, which is basically a kind of Alzheimer's disease diagnosis without amyloid pathology, progressing much more slowly than Alzheimer's disease. The reason why we are not that familiar with those isoforms in the field of AD is that all these isoforms are present. But in some other tauopathies, we only observe 3R tau. For instance, in pig disease, a type of frontotemporal lobe dementia. Or in 4R tau tauopathies, we have corticobasal degeneration or progressive supranuclear palsy, PSP here. And those disorder only includes 4R tauopathies. These isoforms have been known for now more than 30 years. They are supposed to give specific clinical phenotype. For instance, pig disease give behavioral syndromes, whereas corticobasal degeneration give a syndrome where you lose the ability to control one hand and you have a ghost hand. The thing is that the clinical picture depends on the actual brain regions that are involved, not on the actual pathology. So, for instance, we have recently redefined the clinical picture of corticobasal degeneration as the corticobasal syndrome that is most often due to a 4R tauopathy called corticobasal degeneration, but on rare occasions, it can be due to Alzheimer's disease tauopathy. If we are now developing treatments against those proteinopathies, we will need to know which type of tauopathy is affecting which patients, which is not easy. Why? Because biofluids are informing us about a tauopathy, not the type of tauopathy. And amyloid pathology is present in a number of clinically normal older individuals. So when a patient presents with MCI and a corticobasal syndrome, the presence of amyloid pathology doesn't equate to a diagnosis of Alzheimer's disease. This corticobasal syndrome could be due to a 4R tauopathy in the presence of preclinical Alzheimer's pathology that is not responsible for the clinical picture. So the research field needs to develop tools to distinguish between taupathology due to Alzheimer's disease from taupathology due to non-Alzheimer's tauopathies. Right now, it is only possible by performing a brain biopsy or a brain autopsy and measuring the tau isoforms in a brain sample. Biopsies are judged too invasive for patients with mild cognitive impairment, so we can make this differential diagnosis after autopsy only. This is one important set of studies that really inform about how new treatments will need to be different in the different tauopathies, is that the tau aggregates, when looked at with cryo-electronic microscopy, have a very different structure. And the exact sequence of tau aggregating is always in the C-terminal domain, most often includes a microtubule binding domain, but it varies from one tauopathy to another. In contrast with brain measures that are doable with cryo-EM, CSF or plasma measures, biofluid measures, are looking at total tau, tau phosphorylated on some epitopes, most typically in clinic T181, but nowadays we have more and more assays for 231, 217, very different epitopes, but that are barely studied in non-AD tauopathies, and amyloid beta. Several biochemists have tried to measure tau isoforms in the CSF or in plasmas, but they have not proved successful so far. So To address those questions, I decided to leverage the brain samples that we had in Brussels and to conduct a biochemical study specifically separating and distinguishing between aggregated tau and soluble tau, whereas most previous biochemical work on brain samples are only looking at two aggregates. We studied 15 cases with a neuropathological diagnosis of Alzheimer's disease, 5 controls, 5 patients with corticobasal degeneration, so 4R tauopathies, 10 cases with frontotemporal lobe dementia, but 4 of them had 4R tauopathies, 6 of them had 3R tauopathies, and then 5 cases in which pig bodies were observed at postmortem, and pig bodies only contained 3R tau. All the 80 cases are either BRAC5 or BRAC6. All the control cases are BRAC1 or 2, so we have no overlap. And all cases were analyzed with a postmortem delay of less than 8 hours. So let me walk through these results with this first graphic. So what we're showing here is the ratio between 4R and 3R tau absolute concentration in the brain sample. This has been conducted in insoluble tau, so in the two aggregates. And what you see is a logarithmic scale. So basically, when you see 2, it means that this specific subject had 100 times more 4R tau than 3R tau. The 2 subjects here in green have a mutation in the gene coding for tau, P301L, which is giving a 4R tauopathies. And what you can see is that in blue, you have the CBD cases, and in green, the FTLD4R tau cases. And indeed, we find way more tau, way more 4R tau than 3R tau in those cases. In contrast, you see here in yellow and in brown, pig disease brains and FTLD with 3R tau. And in the middle, around 0, so basically 0 means there is as much 4R tau than 3R tau in these aggregates. You have the control cases and the AD cases. They're both on the same level, which makes sense. We know AD is a mixed tauopathy. You see here the absolute quantifications, and we have significant results. That was all expected from the literature, but we could see that our mass spec experiment was working fine. On this line now, you see the same analysis conducted on the same brains. They are presented in the same order. But this experiment has now been conducted on soluble tau. And we were completely unable to separate 3R from 4R tauopathies from AD. In the soluble sample, the quantity of 4R tau and 3R tau is the same. And in these samples that are coming from the brain, we have tau concentrations that is around 10-fold higher than in CSF, almost 100-fold higher than in plasma. So if we are not able to distinguish between 3R and 4R tauopathies by measuring isoforms in soluble brain extracts, in my opinion, it will be impossible in biofluids, either CSF or plasma. We conduct a next experiment measuring the messenger RNA, so by qPCR, to measure tau production in all these cases. And when we are trying to measure 3R isoform separately from 4R isoform, we observe similar levels of production in all these conditions. So the production of 3R versus 4R tau is not modified when a patient suffers from a 4R or a 3R tauopathy. It's the same for both isoforms in pig disease or corticobasal degeneration, including in patients with mutation in the MAPT gene responsible for 4R tauopathy. So these individuals are producing the same level of 3R and 4R tauopathy, of 4R tau. Now, those results are basically negative results. To me, they are pretty important for the field because they are kind of telling the field it won't be possible to distinguish in plasma or CSF 3R from 4R tau isoforms. So we need another strategy. And other teams have been very successful at identifying in plasma and CSF tau post-translational modifications. but their work has mostly focused so far on phosphorylation. By using mass spec, we can actually measure different types of post-translational modifications. And so we repeat our experiment comparing aggregated tau and soluble tau, measuring all the different types of post-translational modifications that we could observe. So this is basically the method. The part A is the part I had just presented to you. It's using absolute quantification with aquapeptide to measure the isoforms. The second part is the post-translational modification, identification and quantification, phosphorylation, but not only acetylation, ubiquitination, methylation. Here are the main results in aggregates in insoluble tau. And these tables are difficult to read, and I'm going to walk you through a first time, and then I will have figure to illustrate those. And if you look here in pink, you see post-translational modifications that are only observed in Alzheimer's disease. And we have ubiquitination on the lysine 311 and phosphorylation on the serine 262, which is within the microtubule binding domain, so in the C-terminal portion of the protein. If you look in blue, we have post-translational modification that we only observed in 4R tauopathies, in CBD, and not in the other tauopathies. And then in orange, post-translational modifications that are observed only in 3R tauopathies, FTLD 3R here. And as you can see, none of these modifications are typical phosphorylation sites from Alzheimer's disease. You see now the same results, but in the soluble brain fractions. So here we have identified way more post-translational modifications, 42 instead of 20. And we have more pathology-related modifications, so modifications that are only observed in disease brain, not in control brains. We find, again, the same type of modifications in Alzheimer's disease, but the phosphorylation on serine 262 is accompanied by an ubiquitination 5 amino acid later on lysine 267. I'll come back to that in the graphic. That would be easier to explain. After observing all these post-translational modifications, we wanted to inquire in the soluble tau if any of these PTM was associated with the ratio of 4R on 3R tau in the aggregate. So basically, could any of these PTM inform us in the soluble fraction about what is happening in the aggregates? What type of isoforms are actually aggregating? And correcting for Banferini, we could identify 4 PTM of particular interest. On the left side of this graphic, you see 2 ubiquitination sites that are strongly associated with an R around 0.5, or above 0.5. So explaining more than 25% of the variance of the amount of 4R over 3R tau. So these modifications are basically mostly observed in individuals who have more 4R tau aggregating. On the opposite side of the graphic, you see 2 modifications, one acetylation, one phosphorylation, occurring in individuals with more 3R tau aggregating. But those PTM were measured in the soluble tau fraction, in the soluble brain fraction. So potentially, we can target them with assays in CSF or in plasma. Here you see the graphic. So in black, you have controls. In purple, you have AD. And as you can see here, ubiquitination on that side is not observed in AD, is not observed in controls, but it's very high in CBD, and it's moderately high in the FTLD cases with mutation in MAPT, so having 4R tau pathology. It is not observed in pig disease or in FTLD cases with 3R tau. Here you have the acetylation on the lysine 311 that you don't observe on controls. You don't observe in 4R tauopathies, but you do see this modification in individuals with either pig disease or FTLD 3R, so 3R tauopathies, to the same level than in Alzheimer's disease. But this analysis correlating with the type of isoform aggregating in the brain did not allow us to identify specific modifications of Alzheimer's disease. So looking back at the data, have we observed modifications that were only present in Alzheimer's disease? The answer is yes. We observed 3 ubiquitination sites that are completely absent in all the tauopathies that we have studied, that is clearly observed in Alzheimer's disease. In contrast, as you observe here, the typical phosphorylation sites most studied in Alzheimer's disease, like P217 or P231, that I know you are studying in your lab in the plasma, were clearly observed in Alzheimer's disease versus controls. But they also observed in other tauopathies to variable extents. So basically, these phosphoepitopes indicate a neurodegenerative disorder. It is not specific of Alzheimer's tauopathy. It can also be observed in other types of tauopathies. Another interesting analysis was to compare the soluble and the aggregated fractions of the brain. What you see here on the left are tau aggregates. And you can observe tau phosphorylated on 217 in the absence of the phosphorylation on 212. That is almost not observed in any condition. In contrast, when looking at soluble tau, actually tau 217 alone was never observed. It was systematically associated with a phosphorylation on tau 2112 that is rarely measured in vivo studies where you need an assay to measure a specific epitopes. This data, to me, indicates that phosphorylation on tau 212 helps tau to remain soluble. In the absence of this phosphorylation site, tau aggregates, and it's no longer observable in the soluble fraction of the brain, but probably not in the CSF or in plasma. Our hypothesis, more mechanistically, is that phosphorylation on tau 212 prevents tau from aggregating. We had similar observation on another phosphosite, which was phosphatau serine 262, which is within the microtubule binding domain. It was phosphorylated in the tau aggregate, but we could only observe it in the soluble tau when tau was also ubiquitinated on lysine 267, again suggesting that this modification protects tau from aggregating and allows to detect the modification in soluble samples. So in conclusion of this study, in the soluble brain fraction, the absolute quantities of 3R and 4R tau did not differ between Alzheimer's disease and non-Alzheimer's tauopathies. The messenger RNA levels of those 2 types of isoform did not differ either, suggesting normal levels of tau production. Neurodegenerative disorders is a problem of protein elimination, protein clearance, not protein production. Measuring tau isoforms in CSF or plasma, according to me, will likely never allow a distinction of AD and primary tauopathies. It's not a method problem. It's not a concentration problem. We cannot do it in the soluble fraction of the brain. In terms of PTM, we have identified several ubiquitination sites that are specifically elevated in 4R tauopathies, mainly corticobasal degeneration and FTLD4R with mapped mutation. The level of these soluble tau PTMs correlated with an elevated 4R3R isoforms ratio observed in the tau aggregates. So basically, to me, those are candidates biomarkers for CSF and plasma that was not measured in this study. Similarly, we have soluble acetylation and a specific phosphoepitope that was elevated in 3R tauopathies, in PIC disorder, in FTLD with 3R tau inclusions. The level of these soluble tau PTM correlated with decreased 4R3R isoform ratio, so basically more 3R than 4R. Finally, we have some ubiquitinated sites, ubiquitin on lysine 311, on 317, on 217 that distinguished AD from non-AD, unlike the phosphosites. And specific PTMs appear to prevent against to aggregation, so they are potential targets to prevent to aggregation and explore new mechanism, new biochemical mechanism to prevent cognitive declines from occurring in individuals with incipients to aggregation. Now, what are our perspectives? The obvious perspective to me is to use those results and expand them to CSF and plasma. We have way more similarities between CSF and plasma with soluble tau than with tau aggregates in the brain. One challenge will be that in CSF, for instance, a big portion of the C-terminal of the protein is missing or is present at very low concentration. And that makes sense. That's precisely the portion that neuropathologists find in tau aggregates in the brain. So if the portion of this protein is aggregating in the brain, it's more difficult to observe in soluble samples. We have recently acquired postmortem CSF from the same cases. So that's what we will first conduct. It's an analysis in the CSF of the exact same cases that we have included here. In our perspective, we will also include more brain regions. All the data I've presented today was conducted in the inferfrontal gyrus. The reason for choosing this region is that we wanted control cases to be really controlled, have normal tau. And as I mentioned, they were BRAC 102 because at age of death, it's very difficult to have BRAC stage 0 cases. But in the inferfrontal gyrus, we could reasonably suppose that they had no tau tangles. Now we will explore the hippocampus, the infertemporal, the inferfrontal gyrus that will be compared, and we will include the entire AD spectrum from BRAC stage 1 to BRAC stage 6, mostly including BRAC stage 3 and 4 that were not included in our study, to better assess the sequence of all these post-translational modification on the soluble protein before it aggregates. In non-AD, our perspectives are to include a higher number of different tauopathies. I mentioned that some epitopes, some PTM were specific for 4R tauopathies, but it's very difficult to affirm if they are specific for corticobasal degeneration, for instance, that was the main 4R tauopathy that we studied, or from all 4R tauopathies. Now we want to expand those results in PSP, for instance, and compare different 4R tauopathies to evaluate if these PTMs are specific of tauopathies aggregating 4R tau or of specific disorders. Also, these specific sites on lysine 311 is particularly interesting because it's only 5 amino acids after the second portion of the microtubule binding domain that is not present on the 4R tau. So we can identify whether this modification occurred on a 3R or on a 4R isoform. And by doing this, to investigate whether this modification is only observed on 3R tau, or if it's present on all isoforms, but that for some biochemical reason, only the 3R tau aggregates when this modification is present. The final but very interesting perspective to me is to better understand how are PET traces binding on aggregated tau, to better understand the relationship between soluble tau brain biomarkers and neuroimaging biomarkers. What you see here is a very recent study from December 2023 that investigated MK6240 using cryo-EM, so on brain samples. And so what you see basically in gray is the tau protein, or more precisely, the tau aggregates. So the C-terminal portion of the protein that is aggregating in the brains of patients with Alzheimer's disease. And what you see in red is where the traces is binding. And so they could determine that the MK6240 tracer binds between amino acid 351, sorry, and 360. We went back to O data after reading this paper and realized that tau was acetylated on lysine 353 in AD, but not in the other tauopathies, except a few FTLD cases. We were very interested in this observation that is currently unpublished, because in our PET data, we see that all AD cases have high binding with MK6240. However, 4 tauopathies are most often negative, but a few FTLD cases demonstrate MK6240 binding. I just made the literature for this talk and realized that a fluoroscopy study has been conducted on FTLD patients. And it has been shown that it was elevated for patients with mutation outside of exon 10 and not for the other. But I'm not aware of any study like this one in cryo-EM using fluoroscopy to determine where is fluoroscopy binding. Is it on the same site that MK6240? I don't know. It should also be investigated whether lysine 353 is acetylated in some specific FTLD patients. What makes lysine being acetylated there? Is that precisely the ones demonstrating tau-PET binding? Is that acetylation necessary for the tracer to bind? So this is the data we have acquired in Brussels with MK6240, where you see in red all or AD cases or MCI due to AD. And you see here amyloid negative, clinically normal. And so our threshold is currently 12% of voxels positive. And as you can see, we have 25 patients with MCI or dementia, so they're clinically impaired, but they're low A beta. And they're basically all negative, but 2. You see one of the 2 on the image here. It's a patient with FTLD. The phenotype is non-fluent primary progressive aphasia. In the CSF, total tau is elevated and p-tau 181. A beta is perfectly normal. A beta PET is negative. That's this person. We don't have the brain, so I can't tell you if lysine 353 is acetylated or not. But that could be an expectation. And that's also where all this biochemical work can lead to, is a better understanding of what makes PET traces binding to. And with that, I'm very happy you have all listening to me. And we have now 10 minutes for addressing questions on any part of this section. And here is a Brussels thank you for all this data. And here is a thank you to all the collaborators I have in Boston. The floor is open for questions.
Thank you. Thank you, Bernard, for your great presentation. I've already seen many reactions. There is the one from Diego, I think, in the chat. I don't know if he can type in this question or if Yakel can let him talk.
Yeah, he can talk now.
Okay.
Oh, I can talk.
I
Okay.
hear you.
Okay, so the question, by the way, we do neuropathology here in postmortem brains, and we work in tau. So we have some phosphoproteomics and tau analysis going on. So it's very interesting what you showed today in your talk. But I have a question that is a little bit like a basic question. And we wonder about, since we work with the postmortem material and we find the changes, and as you say, the changes that we see are not necessarily reflected in CSF of cases, where does the tau that you find in plasma come from? How does it reach the bloodstream?
It's a good question. So my my expectation, my understanding, but I'm not 100% sure. The tau in plasma is coming from the interstitial fluid in the brain. So basically, soluble tau.
Right, the soluble, yeah.
It does not come from aggregates. Aggregates are not moving, are not releasing tau.
Yeah, but how does it get to the interstitial space? That's the question.
Well,
So
so basically, there are 2 or 3 mechanisms. I think that the main mechanism is basically when neurons are dying, a lot of tau is released in the in the interstitial fluid, and which is the reason why we see elevation in tau pathology in the CSF in Alzheimer's disease. Now, even in the absence of neuronal loss, tau is released in the interstitial fluid because it's excreting from neurons, in part in vesicles, in part freely, which is why it's diffuse to the plasma. Now, we have other work that I haven't presented today showing that part of the tau in plasma is encapsulated, is in basically in extracellular vesicle, in synaptic vesicle, which is probably how it's is transferred from one neurons to another.
okay, thank you very much.
Okay, there are 2 questions in the chat. The first one is from Michael Schwarzschild. So he says, lovely biomarker work, thanks. Might high and low 4R, 3R ratios seen in brain aggregates be preserved in EVs or neuronal EVs, even if the ratios are close to 1 in the general soluble fraction?
That's a nice hypothesis that need to be investigated, but my expectation would that it wouldn't work. So the only reason why it would work is that some portion of aggregates are later encapsulated and go out of the brain. So it would really be an active clearance mechanisms of aggregated proteins. I doubt it happens, at least not much. I think that most of the proteins present in EV, so in neuronal vesicles, are soluble proteins. So basically, they are basically a window on intracellular proteins. And so that's what we observed in the soluble brain fractions. What is soluble is the intracytoplasmic fluid and the interstitial fluids. And in those samples with very high concentrations, we could not observe a difference. The protein is produced in the same amount, so we observed a similar ratio.
Okay, so another question from the chat. What are the therapeutic implications of this work for the MCI and AD field? What are your predictions for the role of AI in drug discovery for these diseases?
So first, the first question, the therapeutic implications of this work. At least I see two main impact. One, on the very basic science, is identifying modifications preventing tau aggregation opens a research avenue to foster those modifications and try to prevent the modifications that are fostering aggregation. Tau aggregates are very closely associated with cognition. So basically, I believe that if we can prevent tau from aggregating, it will prevent neurons from dying. Now, a second development is to have more accurate diagnosis and more accurate follow-up of the disease staging. If we better understand the sequence in those tau PTM and what they indicate, we will better select individuals for therapies and better assess how they respond to therapies. Coming back to the example I gave at the beginning of the presentation, let's think about someone with a corticobasal syndrome and amyloid pathology in CSF or plasma. It's very hard to determine whether that person has AD or CBD without autopsy. If we have better biomarkers telling us this is tau pathology, but this is suggestive of a 4R tauopathy or this is suggestive of a mixed tauopathy, basically Alzheimer's disease. That will really help selecting the therapies that we are trying in this individual. Now, the last question, what are the predictions for the role of artificial intelligence? We need big data. Just the work I presented, we had, I can't even remember the number of PTMs that we identified and that we screened, not only identified, but that we screened. Those are astronomic. Mass spec data delivers a huge amount of data, but PET also, like brain imaging, gives millions of data points. And so AI is helping us to analyze all this. Now, more specifically for drug discovery, I'm not a specialist of AI, but my understanding, getting a little bit out of my comfort zone here, is that we could almost simulate also the drug compound that would be most effective on specific modifications, and by running those, you know, in silico instead of in vivo, because clinical trials, as we probably know, cost a lot of money, takes a lot of time, and so if we could simulate those, it's gonna force the field faster the results to come.
Okay, well, I don't see any further question. So it could be last chance.
I have one.
Okay.
Because I have a couple, but only one for now. I was curious if you actually got a chance to look at any effect or impact or sex in the analysis of the 4R tauopathies. I don't know if you have enough samples, but
so we did look, and we did not see sex effects. But as you mentioned, for most of these tauopathies, except AD, we have 5 cases. So basically, those are 3 males and 3 females, or the opposite. By purpose, we selected them to have a gender balance. And we did not observe significant interaction between sex and the PTMs. Probably, like, not probably because lack of power, because I don't know if there are any or not. But yeah, it is clearly underpowered. One potential, you know, if we are able to develop some assays for these PTMs, is to apply afterwards on huge cohorts using plasma samples that have been biobanked. And, you know, as we have seen now with P2217, it's tens of thousands of individuals that have been tested over the past 2-3 years. It's go very fast because sample plasma is easy to get, and also because it has been biobanked from previous studies, and it can be reanalyzed. So I think that those type of studies are more appropriate to test those hypotheses.
Okay, Akil, do you have any other question?
no, for now. Thank you, Bernard.
You're very welcome. We're very happy to present and to collaborate.
It was great to have you today, Bernard. Thank you again for this great presentation. I don't see any other question for the moment, any other end either. So yes, a final thank you for your talk, and for all of you to be attending this MAPP round. And I'll be happy to see you on the next one. Thank you very much, everyone.
Thank you.